Thank you very much for this kind introduction. In my following presentation, I want to talk about proteomics and remodeling of the heart proteome in cardiac disease. I have no disclosures. The term proteome describes the entire set of proteins within a sample. So, for example, a cell or a tissue sample or liquid sample. Mass spectrometry-based proteomics is the biochemical method used to quantify 3,000 to 7,000 proteins within this given sample. And biological samples are pre-treated and proteins are broken down to peptides and then quantification can be performed. This quantification of so many thousand proteins at once from one sample enables an untargeted and targeted approach to study differences in protein expression levels between samples. So why are we interested in proteins? Proteins are the functional units of the cells with certain biological properties, and those biological properties, they enable proper organ function. And in the absence or dysfunction of specific proteins, this can lead to significant organ dysfunction. In one example, in Duchenne muscular dystrophy, for example, the protein dystrophin is not present due to gene mutation, and this leads to overall skeletal weakness and cardiac involvement with reduced cardiac function. So the aims in research for proteomics and also for the clinical use is hoped to be to improve mechanistic insights of certain diseases and to help discover new druggable treatment targets. In cancer research, it's described that proteomics, for example, identified new therapeutic targets, in this case, as one example, in hepatocellular carcinoma. Proteomic profiling of over 100 samples of hepatocellular carcinoma samples has shown distinct proteomic differences to subtype these hepatocellular carcinomas. And here you see 3 subtypes. And the third subtype actually was associated with the worst clinical outcome with the very worst overall survival. Furthermore, this research group was focusing on single proteins which were higher expressed in this subgroup, and they found higher level of SOAT1 protein expression, which was associated with poor prognosis, and functionally, which promotes growth and migration. This SOAT1 protein, there already exists a pharmacological SOAT1 inhibitor, which is already used in cancer medicine as well, but not in this case. And yeah. Further research, they saw that downregulation of SOAT1 suppresses the proliferation and migration. And so the authors suggest that SOAT1 might become a druggable treatment target in hepatocellular carcinoma in order to improve overall survival, especially in the subgroup 2. So this is just one example that proteomics might be a promise in a new area of precision cancer medicine. So what about cardiac proteomics? The heart proteome might improve, for example, mechanistic insights into myocardial remodeling in different pathologies and in different individuals. It might help to discover new diagnostic or prognostic biomarkers, and it might help to discover druggable treatment targets, for example, to prevent or reverse maladaptive remodeling. In 2017, the first time a proteomic map of the human healthy heart was published, unfortunately only in 3 male hearts, so the female hearts are not yet published. That to come. And the group reported proteomic profiles at different parts of the heart, so in different parts of the ventricles, the atria, the great vessels, and also all the 4 valves. They report regional differences in the heart, which is not surprising, but important if you compare parts of the heart to diseased heart. So they also compared healthy left atrial protein expression levels to left atria of patients with atrial fibrillation, and found differences in extracellular matrix proteins and mitochondrial proteins. But they did not only find differences between patients with atrial fibrillation and healthy material, they also found differences in protein expression between different patients. So they only studied 3 patients. So in regard to prognosis, not much can be said, but as an outlook for the future, if different patients had different prognosis, this could be linked to the proteomic profile as well. We performed a study in patients with severe aortic valve stenosis and severe mitral valve regurgitation. We wanted to study the different phenotypes of pressure overload concentric hypertrophy and volume overload eccentric hypertrophy, and performed proteomic profiling. So cardiac biopsies were taken from the left ventricle, and proteomic profiling was performed, as well as clinical imaging. And so we compared disease-specific differences and differences between female and male patients. Approximately 4,000 proteins could be quantified from the samples, and we could detect large proteins like titin and also small proteins like heat shock proteins. And here protein expression levels were seen that control patients had an overlap with mitral valve insufficiency patients. However, aortic valve stenosis patients almost had no overlap with the control patients. It was an untargeted approach, so we were not in the first place looking at specific proteins. Here in the volcano plots, you can see differently expressed proteins between control material and aortic valve stenosis. In the middle, you see the differences between controls and mitral valve insufficiency patients, and in the left, the right panel, between mitral valve insufficiency patients and aortic valve stenosis patients. So we saw actually protein expression differences compared to controls in 4 categories: the extracellular matrix composition, energy metabolism in mitochondria, cytoskeleton and muscle contraction, and proteostasis. Whereas in aortic valve stenosis, we rather saw changes in proteostasis proteins, and in mitral valve insufficiency patients, rather cytoskeletal muscle contraction proteins. From clinical imaging, we were seeing similar amount of myocardial hypertrophy in both patient groups, and similar amount of fibrous tissue content by cardiac MR imaging. However, a significant difference in end-diastolic volume index, so mitral valve insufficiency patient for sure had larger ventricles than the aortic valve stenosis patients. Looking at female and male differences in patients with aortic valve stenosis, you see in pink protein expression levels which were selectively only changed in female patients, and in blue proteins which were selectively only changed in male patients. And clinical imaging as well showed significantly higher myocardial mass index in male patients than in female patients, and as well a significant higher amount of fibrous tissue content in male patients than in female patients. So looking at the categories, so proteins related to the extracellular matrix, we found upregulated in aortic valve stenosis patients and in mitral valve regurgitation patients compared to the controls. And in clinical imaging, like I said before as well, fibrous tissue content was increased compared to the reference range. However, looking at single proteins, we saw different proteins to be upregulated in aortic valve stenosis than in mitral valve regurgitation. In aortic valve stenosis patients, for example, fibrillar collagens like collagen 1, 2, and 3 were upregulated, whereas in mitral valve regurgitation patients, more enzymatic proteins and proteins of developing arteries and endothelial cells were upregulated. In regard to female and male differences, again here in aortic valve stenosis, the male hearts showed higher fibrous tissue contents than in female hearts. And in proteomic profiling as well, we saw only specific male upregulation in male patients and no specific ECM protein upregulation in female patients. So antifibrotic therapy in clinical trials often failed, and perhaps differences between individuals, difference between female and male composition, and differences perhaps also in cardiac etiologies might be the cause for this. In a very recent publication, the cardiac proteome has been compared between human and 5 animals in order to see whether protein expression levels at a healthy state are comparable or not. And they were actually looking as well at different disease-specific proteins. And just as an example, ARVC-related genes and proteins that we're looking at. And they saw the best match actually for human expression levels in pigs, and and not a very good match with mouse or rat, for example. So these types of publications might be helpful in order to plan the most successful animal model in order to be then converted to human data as well. Looking at cardiac metabolism and proteins related to cardiac metabolism, we saw that in aortic valve stenosis patients, proteins of cardiac metabolism were reduced, markedly reduced, and in mitral valve insufficiency patients, there was only a slight reduction. Again, female and male differences we saw with the least changes in female patients with aortic valve stenosis. Interesting to highlight would be a protein called TSPO, which is a translocator protein of the mitochondria, which were upregulated in aortic valve stenosis patients and downregulated in mitral valve insufficiency patients. From animal studies, it is known that the prevention of upregulation of TSPO was associated with better cardiac function, more preserved ATP production, and reduced oxidative stress. And they even suggested TSPO as a druggable therapeutic target in order to prevent heart failure, which then, going back to the human, would only be adaptable for aortic valve stenosis patients and not necessarily for mitral valve insufficiency patients, because they already had a reduced level of TSPO. Proteostasis was another category we were looking at. So proteins related to protein synthesis. And there again, we saw a remarkable reduction in protein synthesis proteins in aortic valve stenosis patients, and only mild and mitral valve insufficiency patients. And even more remarkable was that we were actually only seeing this reduction almost exclusively in female AS patients. And since in these aortic valve stenosis patients, we were seeing the remarkable sex differences in myocardial mass and myocardial hypertrophy, and protein synthesis is necessary for cardiac hypertrophy. This might suggest a molecular mechanism why female patients with aortic valve stenosis might be might be safe for excessive hypertrophic response in aortic valve stenosis, perhaps. Cytoskeletal proteins were again upregulated in aortic valve stenosis patients and in mitral valve regurgitation patients, but again, different proteins were upregulated in AS and different in mitral valve regurgitation patients. So in aortic valve stenosis patients, there were more actin bundle proteins which were upregulated in mitral valve regurgitation patients, more cell matrix adhesion proteins. So suggesting that the mechanical load actually leads to differences in cytoskeletal proteins and also according to the mechanical load and the need for different functional proteins. We also possessed a targeted approach in our patients with aortic valve stenosis. So aortic valve stenosis patients show differences in the degree of hypertrophy despite comparable AS severity. So not only differences in hypertrophy between female and male patients, but also in between female patients and in between male patients. And it's known from the literature that event-free survival is significantly better if less hypertrophy is present. And we know that female patients have less hypertrophy and less fibrosis and lower NT-proBNP levels. And we also know that the survival of female AS patients is better. So this suggests that female or male hormones might play a role in this hypertrophic response. And in mice, it was published that blocking testosterone with finasteride, which is a dihydrotestosterone blocker, and dihydrotestosterone is the active metabolite of testosterone. So blocking this testosterone attenuates cardiac hypertrophy and left ventricular dysfunction in mice with transverse aortic constriction, so with aortic valve stenosis mice. So we were also measuring dihydrotestosterone in our patient with aortic valve stenosis in order to correlate this to left ventricular mass, fibrous tissue content, and function. And we found a positive correlation of dihydrotestosterone with left ventricular mass and with fibrous tissue content, and an inverse correlation to left ventricular ejection fraction. And looking at the proteomic profile, we were specifically looking for a protein which is known to be influenced by dihydrotestosterone and which is already known to be pro-hypertrophic, the protein MSN. And we were also finding a positive correlation between dihydrotestosterone levels and MSN. And we were also studying protein bundles of vimentin, so pro-fibrotic proteins. And this we're still performing and looking for pro-hypertrophic and pro-fibrotic proteins in relation to DHT and MSN protein. And additionally, we could see that higher levels of dihydrotestosterone before aortic valve replacement in these patients with aortic valve stenosis was actually associated with a worse outcome after AVR, looking at ventricular function and fibrous tissue content. So the higher the dihydrotestosterone level was before operation, the lower the function was after operation, and the higher the fibrous tissue content. So I want to conclude that cardiac proteomics might help to improve mechanistic insights into myocardial remodeling in different pathologies and in different individuals and between female and male patients. And possibly, a lot of research has to come, but it might help to discover possible treatment targets to prevent or reverse maladaptive remodeling. Thank you very much.
Thank you so much, Sarah. Since I can't see any entries in the chat, I'm asking the first question. So Sarah, obviously we are lacking a little bit behind our colleagues from oncology, I would say, who did molecular characterization of the diseases a while ago. So what is your estimation in terms of drug targeting, developing new compounds, and so on, from the information you can see from your patient groups in structural heart disease? How big is the avenue?
Well, I think the biggest problem is that we don't have the outcome like in cancer medicine. So in cancer medicine, you really have a strong outcome. And in cardiac disease, it's more difficult, I think, to get the strong outcome. It's many years after. I mean, all these aortic valve stenosis patients, they're doing fine. So they are 65 and they received their aortic valve replacement, and now it's good for now. Definitely. The remodeling part is interesting. However, it's probably not influencing survival in the next 2-3 years. So we probably have to focus on really, really strong outcomes to organize our studies, to plan our studies in order to really find interesting things.
So is it then hopeless or to do this kind of research?
No, not
I'm
as
asking you a little bit provocative, of course.
No, but I mean, they started many, many years ago, and in cardiology, proteomics, well, is like few years.
Okay, thank you so much, Sarah.